Now I'd like to hand over to Erica Nielsen from South Africa.
Hi.
A PhD student at Stellenbosch University in South Africa. And she's going to talk about spatial planning for the future using molecular and modeling tools to identify conservation hotspots. Erica, the stage is yours.
Thank you. I hope everyone can see my screen.
Yep.
Thanks for the introduction. And I'd just like to thank IMCC for organizing, and especially for the transition online, as I think definitely this being a climate change session, it really is the future for conservation and climate change conferences especially. So with that being said, welcome to my talk. I'll be talking about how to assess vulnerability using some exciting new tools and using that to implement conservation plans. So MPAs have been around for quite a long time, and for most of their existence, they were mainly focused on regulating fishing, as back then we generally didn't see long-term climatic change as posing a threat to marine systems. However, we now know better, and thus MPAs are shifting their objectives to include climate change and species responses to climate change, whether that be via adaptation or shifting their distributions. So when we think about designing MPAs with regards to climate change, the general roadmap is to, number one, assess vulnerability, and then from there, design or adaptively manage your MPA to promote resilience to climate change. So this seems pretty straightforward and easy. However, when we think about actually doing this, things can get really complicated really quickly. So there's multiple different ways to assess vulnerability over different spatial and temporal scales, and we can also do it from the population all the way up to the ecosystem level. So it's really no surprise that in a recent review by Wilson et al. 2020, they mentioned that one of the greatest barriers to including climate change adaptation into MPA design is that there's very few case studies on how to actually do this. So that's why I'm here today to provide a framework to assess vulnerability using a South African rocky shore case study in which we compare resilience between not only 3 different species, but also using 3 different vulnerability metrics. So here are our study species. We chose them as they have the largest molecular data sets for the region, and they also can capture the different rocky shore ecological niches as much as 3 species possibly can. As I mentioned, this study was conducted within South Africa. It's one of the most biodiverse countries in the world, and it has these really beautiful environmental gradients, as we can see here in sea surface temperature. Here's the distributions of the 3 study species, and here we have our sampling regime within the study, including either 13 or 14 sample sites per species, and from there we conducted molecular analyses. So our first vulnerability metric is climatic stability, and to generate this we ran species distribution models at 5 different time points, actually starting in the past at the last glacial maximum 21,000 years ago, all the way into the future at 2,100. And so these models were run for each species individually, but using the same 4 environmental predictor variables, which were found to be uncorrelated for all time points and all species. So here what we were looking for was to identify stable populations through time, as they're expected to be more resilient to future change. Our second metric is genomic diversity, and so from those sample sites we took tissue and then extracted the DNA and then ran that through high throughput genomic sequencing, where we looked for SNPs throughout the genome. And so a SNP is basically just a mutation, and we can look at the amount and the frequencies of these mutations to get different population indices. So the one that we focused on here was expected heterozygosity, or HE. It's a very standard metric, and so what we were looking for here was to identify highly diverse populations, as there's more genetic material for selection to act on, and they also have larger population sizes, both of which should increase resilience. And our third and final metric is genomic offset. It's probably the least well known. So to create this, we ran gradient forest models, which use a machine learning algorithm, and they were originally used to map species composition in space. So in this figure here, this is the Great Barrier Reef, and the different colored areas represent different species compositions based on the model. But we know that species doesn't respond as a whole to the environment, but rather different populations within a species have different trajectories. So a great example of this is by Stanley et al. 2018. And so they looked at 5 different marine species across the Northwest Atlantic, and using genetic data, they consistently found that within each species, there was a warm adapted and a cold adapted population. So ideally, we would run these models at a population level to best understand how a species responds to environment. So that's basically what we did here. So we used these gradient forest models to map not species composition in space, but genomic composition in space, using 8 environmental predictor variables. And so what we see on the bottom here is an example from Keller and Fitzpatrick, 2015. So these models use turnover functions, which is shown in the plot on the left. So looking at how much the SNP frequency changes over an environmental change. Those are combined for different environmental variables to then map our genomic composition in space, which we see here for a tree species in North America. And so the coolest thing about these models is we can create these maps at different time points, and then from there we get our genomic offset. So here we wanted to find areas of low offset, as the genomic variation has to change the least to track future environmental change. So using the same example, that would be site B here. So looking at the plot on the right, it sees the same environmental change as site A on the x-axis, but the genomic composition has to change much less, which is shown on the y. So here what we were looking for was to identify low offset populations, as they have to adapt less to track future environmental change. So finally, we had to map each of these metrics. So to do that, we created binary presence-absence maps from the species distribution models, and then only mapped if the species was present at each of the 5 time points. Genomic diversity, we first had to interpolate our point data over the entire planning region, and then we created low, medium, and high-ranking groups, and then only mapped if the area fell into the high-ranking group. Genomic offset followed similar protocol, except now we only mapped if the area fell into a low-ranking group at both time points into the future. All right, so what did we find? Here are the resultant maps. Broadly, what we can see is there's quite a bit of variation, not only between the different vulnerability metrics, but also between our 3 species. And so these maps will go into a spatial planning tool, such as Marksan or zonation. But for now, what we can do is try to look for areas of overlap between all of these maps. So the first area that pops out is around Cape Town and False Bay. And then the second one is around Cape St. Francis and Port Elizabeth. And so we can compare this to previous studies in the country, and quite interestingly, these same 2 regions were identified as refugia at the last glacial maximum solely due to sea level change for 2 different lineages of a clipfish. So that's shown in the green and the pink circles there. They've also been highlighted as hotspots for endemic marine invertebrates. So looking at this plot here, on the x-axis we have distance around South Africa's coast, and then on the y we have the number of endemic species. And so these 2 spikes actually correlate to these same 2 areas. Also, out of interest's sake, I wanted to include a socioeconomic metric of vulnerability. So here is the expected population growth pressure within the country over the next 3 decades. And we can see that our two hotspots are actually expected to see quite an increase in population growth. And finally, here is our current MPA network within South Africa, and we just implemented 20 new MPAs, so that's quite exciting. However, when we tried to look for our hotspots that we identified here, they lie within current gaps in our network. So this highlights how these two areas may be seen as priority areas for future conservation efforts, as they've been shown to be ecologically and now evolutionarily important. So in conclusion, here we provide a framework to assess vulnerability, and we found that the different vulnerability metrics as well as species offer different resilience hotspots. However, when we looked for areas of overlap, our hotspot regions actually corresponded to both historical refugia and ecologically important areas. So this highlights how there might be some exciting new opportunities to investigate surrogates of vulnerability, as genomic data is quite time and cost intensive to generate. And this is especially true when we think about kind of taking this to the next level and assessing ecosystem level vulnerability, which is very much still needed going forward. So with that, I'd just like to say thank you, mainly to my supervisors and co-authors, everyone who's helped me along the way, and thank you for you all for listening to me today, and I'm happy to take questions.